This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 201, recorded on June 21st, 2019. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Today we're recording in San Francisco, California. We are at ASM Microbe 2019. And joining me here in Vivo, Michael Schmidt.
Hello, Vincent.
Nice to see you.
It is good to see you, too.
Normally we don't.
Normally we turn off our cameras when we communicate with one another because it takes up bandwidth, and we want to make certain that the sound is as good as it can be.
That's right. We have a guest today, and he is returning about 1 year later from his last appearance on TWiM, which would be episode 180. He's a professor of biology at the University of Puget Sound. Mark Martin, welcome.
Hi, Vincent. Hi, Michael. Hello to you and your microbiomes.
We hope they send their greetings as well, but not too close.
How is Tacoma these days?
Oh, it's quite lovely. I mean, for me, I love the fact that there are some areas there where there are huge amounts of hydrogen sulfide producing organisms, and we have wonderful thiotropic symbioses. We call that the aroma of Tacoma.
You mentioned that last time. You said you were thinking of working on it, right?
I love it. And I am going to be taking some samples this summer from there, and it's actually relevant to today's topic. Some of the Merithrix, which appears to be the major organism that we find in these sulfide seeps, they are covered with other bacteria. So once again, the microbial world is all about interactions.
Mm-hmm. Interactions like those on TWiM, right?
That is true. We've certainly been interacting here at this meeting, running between poster sessions and talks, and even down in the exhibits. We're bumping into old colleagues, new colleagues, and just people who recognize our voices from TWiV and TWiM and TWIP and all of those things.
I've been calling that Twix.
Yeah, I like Twix.
Yeah.
But there is a candy called Twix, you know.
And we're worried about the copyright police who will come and get us in the middle of the night.
If you like our work, by the way, consider supporting us. You can go to microbe.tv/contribute. A couple of ways you can do that. We'll talk more about that later. I asked Mark what he wanted to talk about. Last time we talked about Mark and his teaching and his work, and he said he wants to talk about great microbiology papers for first-year students. And he sent a bunch, which we'll go through today. So if you're a teacher of undergraduate microbiology, you should listen because Mark has some wonderful suggestions.
Before you go there, I wanted to ask, are these for majors or non-majors?
I love the way you asked that question because it shows you're from a larger institution.
We are.
As I went to UCLA as an undergraduate. Sid Rittenberg was my first microbiology professor.
Really?
That's correct. Okay. Did you know Gary Wilcox and Bill Romig? These were all buddies of mine. So anyway, the problem is at my institution, there is only one microbiology course, and it is a course taught primarily to juniors and seniors. But I have a plan. My plan is to colonize other courses. And I've had some success in doing so bit by bit because I believe, as many other people do, there should be much more microbiology in the curriculum early and often.
I agree. I think we should start introducing it in middle school, but that's just me.
And I know Von Cooper's been working on things like that with certainly high school students, and I agree completely. But one of the problems that you have is the people that come to you, say, in your first year biology class. And to get to your direct question, many, many students at my institution will end up taking my introductory course from outside the major as a breadth requirement. And I will not change it from the way I normally teach it because I also have pre-biology majors in there.
You should be commended for that because I think that we should never water down the discipline. I think if the discipline is delivered properly, Microbiology is universal.
I agree. Abigail Salyers and I used to say first evolved, last extinct.
That's good. Abigail was a force of nature, and I still value many of her wise words and counsel. Abigail and I, when the society held its meeting in Toronto in 2007, Abigail and I were on a mission to go evangelize microbiology, and so we had to go to an art show. And we were on a panel with artists doing what Dr. Martin does so well, using bacteria as the medium to deliver art form. And Abigail was out of her element. She was very much in her element, talking science, but interacting with the artists and using bacteria to illustrate on Petri plates. She was very uncomfortable because she was concerned about germs spreading, and she didn't use the word germ. She used, but the audience kept using the word germ, and she was a bit uncomfortable. But we got her to evangelize the discipline, and the artists got it in the end. They understood the value. They understood the importance of infection control and being safe when they worked with the microbes to make certain that it wasn't hazarded about. And it was overall a really fascinating session.
When I've had that and I do this with freshmen as well as when I suppose I should say first-year students nowadays. The language is always changing on me, Michael.
Oh, yes.
But in any event, I often go and I put on what I call Lux Art shows, where I encourage people to use cultures of Photobacterium leonothi as the medium. And I'll often hear from people, oh, that's gross. Oh, those are germs. And I'll say to anyone with artistic skills, so when you paint with acrylic paint, do you put that in your mouth? Do you wash your hands before and after? Because I would actually argue that the acrylic paints are more toxic than that particular strain of bacterium.
Well, certainly making them are.
Right. So, but you glove up and you do the painting, and it's part of my overall plan. To get back to what Vincent was asking, I wanted to infect the first year courses with more microbiology. And I'm not putting down anyone when I say that. I mean, to a carpenter, every problem involves a hammer and nails, and I'm the only microbiologist so at my institution, so that's a big deal to me. So I thought, could I take advantage of the fact that we want to put more mathematics in our first year curriculum, statistics specifically? So John Lennox, who I don't know if you know.
I know John.
He had taught me many, many years ago at an ASMQ about using Pseudomonas putida as a biofilm, a very robust biofilm former. And I used 24 well plates, and I came up with a way where you could get 6 replicates for each condition in a 24 well plate. And then that way the students could learn a little bit about biofilm formation because the procedure of crystal violet staining and extraction is fairly straightforward. And then we would ask, what could we do to study what affects biofilms? I always start with an observation that I had made with that organism is that alpha amylase will inhibit biofilm formation. And then I also show them a wonderful one-page paper from Nature, and it is by Cynthia Whitchurch, showing that DNases will interfere with biofilm formation in Pseudomonas aeruginosa. And so then we talk about its relevance to health and all the rest. So the point that I'm trying to get at is most first-year biology introduction to microbiology is, can you streak a plate? Can you spread a plate? Can you count a colony? Can you use P-glow? That's the big deal.
It's mechanics
agree.
rather than the wonder.
I agree.
I think the entry-level courses need to reinforce wonder.
I agree.
And that's what will turn on the light and get those students interested in science. It doesn't matter if it's microbiology, but get them into critically thinking about life. Mm-hmm. I mean, that's what college is, after all about, critically thinking. And if you can frame your introduction to anything with wonder, that's generally a hit show.
That reminds me so much of some of the talks I've given, Michael, that you haven't been at, because I make that same point. So great minds with but a single thought. So
Can I ask you?
whatever.
Hold that thought. I'm sure it's good. Can you give a first year undergraduate a microbiology course, or do they need prerequisites? Coming from high school where you have no idea what kind of science they've had, like biochemistry or chemistry or biology even. Or are you suggesting sneaking a little bit of micro into first year courses?
Absolutely.
The latter.
Yes. And we also, I'll have students that are, say, an economics major that are taking that first year course of mine as a breadth requirement, and they don't have any of that background.
But they do understand exponential growth.
I'm not so sure about that.
Economics majors should understand exponential growth.
Do you teach,
otherwise, they're not going to be good.
What is this course you're referring to that you teach first year?
It's called The Unity of Life.
Okay, it's a biology course?
Yes, it's Introduction to Cell and Molecular Biology.
So they need that to take a micro course, right?
I need it for life.
Yeah.
Yes, that too.
So my point is there were a couple different things about microbiology I wanted to be in that course. And so I was able to put in the biofilm module. I tested that with seniors. I asked them what they thought, how that would work with first-year students. So I got a lot of good feedback. And now it's accepted. And one of the things that's been fun, and I say this to you because I know you'll love this, we discovered that red wine inhibited biofilm formation in that particular system. And it is truly wonderful to sit down with the students and say, all right, what do you think might be inhibiting? What's in red wine? And for once, they whip out their phones, and I'm happy.
And they're going to town.
Right. And then we're actually able to say, well, if it's the alcohol, what is the percentage? How would we set up something in these little 1 milliliter
Very nice.
sets? So it works pretty well.
Very nice. All right, now we're going to use Mark's show notes because he wrote these show notes. Very good.
That was good.
The first one, there's an expression I came up with, OMG. Let's talk about OMG. What is that? It's actually a hashtag OMG, right?
Hashtag OMG for overwhelming microbial greatness.
And you made a badge to go with it.
Yes.
Oh, what you have there. Now, describe what's on this badge.
Well, I have to say this is a wonderful piece. Sarah Atkins at the University of Alabama did this artwork for me. And I had been bandying about the term OMG and overwhelming microbial greatness, just as I do with things like quality quorum. And I was afraid they would get away from me. So I thought as soon as I made commission artwork with it, then it's more mine.
So the individual on the left, I'm guessing, is von Leeuwenhoek.
Yes,
it is. Excellent.
And the individual on the far right looks to me to be the former, one of the former subdirectors of the Office of Technology and Science Policy.
That's correct.
Joe Handelsman.
That's correct.
Is the guy in the middle Carl Woese?
No, don't you wish?
He's not on this?
I'll give you a hint. No, I wish he was, but I didn't choose any of these. Sarah did.
Sarah did.
I said I want 3 men and 3 women that really speak to you. And so I was delighted about Leeuwenhoek, and I was delighted to see Joe. But in the middle, here's a hint, penicillin.
Well, Fleming.
Yeah, that's Fleming.
With the bow tie.
Bow tie, I guess, yeah. Okay.
In between Leeuwenhoek and Fleming is one that I think we need to talk about more and more in classes. That's Fanny Hesse.
Oh, the Agar lady.
Exactly.
Do you know the story of Frau Hesse? I learned this story probably from the dinner party guest when I was a graduate student at Indiana. So I'll just digress for a moment since Michelle's not here to hit me.
Or Elio, right? Or Elio usually hits you too. Right?
Yeah, Elio hits me. So Frau Hesse, this wife of Koch's technician, Herr Hesse, was trained at the Sorbonne in Paris. She was a very good cook, and she was making a very fine and elegant dessert, these glacés that use this extract of seaweed, agar, and she served the strawberry glacé at one of the dinner parties. It's when faculty got together with their technicians and had elegant dinners after work, and she had this, and they're all eating this wonderful dessert, looking at this shiny gelatinous-like substance, and they had their aha moment. Said, could we use it? Because at that time, what the lab was experimenting with was gelatin to create solid media. They were using potato starch. They were using all sorts of things to see if they could develop a solid medium from which to get isolated colonies. And so Hess talked to his Frau Hess, and voila, we have agar-agar solidifying agar, or solidifying bacteriological media. And that literally revolutionized our discipline, because for the first time, we were able to isolate individual strains of bacteria. Now, think about if we were never able to get to that aha moment, we would have been doing the microbiome 100 plus years ago, because we were never able to isolate single species or single organisms. Because of that, we would have had a very different discipline today, because we would have been exclusively working on communities the whole time.
It's an interesting way to look at it. And I've often told people that microbiology is at the center of most biological science. Imagine the field of genetics if microbes weren't available.
Oh, it'd been very slow.
Right.
Peas grow slow.
Mark, what is the viral equivalent of the story the assay that Michael just told us?
Oh, now you're going to be,
It begins with a P and ends in an E.
What assay allowed, as Michael said, the isolation of single cloned viruses?
Oh, no. Now you put me on the spot. Please tell me.
It's the Plaque assay, of
Plaques.
course.
Okay.
You must teach this at some point.
I do.
Right?
I do teach
that allowed
plaques.
us to purify and clone viruses, but we now know that it's not really a clone, because as soon as they grow, they diversify into a quasi-species. But similar advance, right?
Yeah. Right.
But using agar, of course, which came
soft
from
agar.
bacteria. Yeah.
And that's necessary for a plaque assay as well, which is very interesting to think about. Now, I can twist this around and say, as you alluded to, that perhaps that approach of looking at single isolates and single colonies has its problems. But I defy you to think that we wouldn't have lost huge numbers of lives.
Oh,
think about,
without a doubt.
Right. So there was a time for it, is my point.
No, and I don't think we were intellectually capable of looking at the complexity of life 150 years ago
Agreed.
the same way we are able to do it today.
So who else do we have on this?
Well, next to Fleming is someone who's relevant to what you're discussing. That would be Esther Lederberg.
Oh, Esther.
Replica plating, right?
Yes.
Yeah.
Phage lambda, right?
Yeah.
And the gentleman with the wonderful hairstyle is Sergei Winogradsky.
Okay.
Oh, yes, Serge.
Interesting collection.
Yeah, I did not pick them. Sarah did. Sarah Atkins is a fabulous, fabulous artist in general, but loves doing microbial art.
She's a graduate student.
Yes.
Great
She's
job.
never heard of
Sarah.
C.B. Van Niel.
These are all
These
heroes
are
of mine,
hers.
but these are what she
So
picked.
one would say we should do this for viruses, except the hashtag is just not as good. IMG is just awesome, right? Because it's oh my gosh.
Well, viruses are microbial.
Well, yes, I guess.
Vincent, can I leap in and defend? You remember, I took virology. I took it from Arnie Burke, okay, at
Good
UCLA.
teacher. Good teacher, yeah.
Right. So I appreciate viruses, and the hard part is it's very hard in my course to slide any of that in other than phage. And it's something you and I have to talk about, because I think it's really important in fact, I just thought of you. Didn't you just tweet about the fact there are now primer-independent
DNA
polymerases?
Yes.
Something that now I have to change the way I teach things.
Well, you could, I'll tell you what, I'll come for a year as a visiting professor and start a virology course.
I would love that.
And then you can keep it when I leave.
A sabbatical for Vincent. Film at 11.
So we talked about having just one microbiology course, how you infect kids. You talked about the biofilm experiment as
Right.
a way of infecting them with excitement.
Yes.
And then you talk about how first years are a diverse lot, and some really get into research and others don't. Tell us about your experience there.
Well, I mean, some students are just interested in going through whatever hoops are available, or they'll decide it's not interesting. It is no surprise to you that I tend, when I teach, toward the flamboyant, and I do that for a reason. And it's not because of my personality. It's because you have to get their attention, and you cannot fake your true love of a discipline. And students always respond better to your enthusiasm.
Absolutely.
If you're not interested,
Absolutely.
Michael, in a
If
topic,
you're bored, you're bored.
it comes across.
No, passion can really make a difference.
And so when I will tell them something, and a really good example of this, and it's relevant to one of the papers I talked about, and I do this in my first year class, I ask them what the two most important enzymes in the biosphere are. And they invariably tell me DNA polymerase.
And the other one?
Alcohol dehydrogenase.
Well, depending on the grouping, yes. But then I tell them what is DNA made out of, and I lead them bit by bit to carbon fixation by rubisco and organically available nitrogen via nitrogenase, both of which are bacterial enzymes.
Mm-hmm.
And this generation definitely needs to hear about rubisco because we're facing a climate crisis, and we need to know how to fix carbon so we can get our CO2 part per million level down. We need to understand fundamentally how to exploit rubisco better so that we can fix some of this carbon we've unfortunately burned and put into our atmosphere.
And it is relevant because you need to find things, especially for first-year students, which takes us back to our topic, that really grab them. Now, by the time I see juniors and seniors, they've taken usually genetics and cell biology, and part of that is where they dissect papers. And that's fine.
So they're critically thinking at your institution at the undergraduate level. They're not binging and purging.
Well, I'm withholding my comment on that. But
you don't want to get you fired.
No, and it's hard to do at this stage. I would like to think, but I'm just looking at Elio, looking at me, who's not here, but he's kind of giving me this, and so I giving me that eye. So I'm going to be better. My point is that they've learned how to analyze many types of figures and kind of chamois how to analyze the data. The way that I like to teach it are positive and negative controls, and they're really important. The problem that I find is that if I dump a microbiology paper on a first-year student or an advanced microbiology paper, even on my juniors and seniors, they don't have enough background. There is a language to every subdiscipline.
Of course. How many of your science majors want to go to medical school? What fraction?
Or just professional school, dental school, medical school, pharmacy school?
So we graduate every year about 54 or 55 biology majors. Remember that the University of Puget Sound is comparatively small. Of that 54 or 55, I would easily say 10 or 12 are interested in a medical path. Another 10 or so are interested in a graduate program of one form or another, especially if I've infected them to some degree.
Okay.
How many want to become brewers? Because out west, there are lots of microbreweries.
No, that's just funny on several levels, microbrewery, but there you go. I would say they're only now starting to get to that. A lot of them are trying to make adult fermented beverages, which I don't look at very carefully. Age, under 21.
Oh, yeah. Okay.
Freshman, yeah.
Yeah.
All right. You sent us a bunch of papers. Let's go through them because they're awesome.
They're awesome papers.
First one, it's a opinion by Elio, which was published in Microbe in 2009. Paradigm shifts, paradigm drifts. What do you like about this?
I like it because it frames my microbiology course and it enables students to find – well, the way I describe teaching a little bit, Vincent, is I want you to imagine a tree without leaves. Think of a holiday tree, a Christmas tree, a Hanukkah tree, whatever you want to call it, and it's just got the branches on it. So you have to have that first before you put on the ornaments or before you put leaves on, whatever it is you talk about. There needs to be kind of an organization that you can integrate into. Otherwise, what you have is a bunch of 3x5 cards that you riffle through right before exams. And I do not think that that's true learning.
No.
I think that's memorization. And I would like to think, my goal is when my students look at my course a year afterwards, that they take certain ideas away with them that are useful to them.
Yes.
So Elio's wonderful essay, and I wish upon wish upon wish that we could have an updated version of it, breaks things down into 5. And what I tell my juniors and seniors when we talk about it is it's important to kind of look at things that I'm presenting and figuring out where that information would integrate. So when we talk about planetary prevalence of microbes, there are lots of examples of that. And one thing that I find really works is when students are interested in something, they remember it better. So I'll tell them within the confines of that area what really sticks out in your mind.
What do they say?
Oh, they'll say things, the numbers, and we have to have the argument about is 10 to 1 really true, right?
It's now 1 to 1.
What about microbes comprising half the total weight of living things?
Yeah.
Is that right?
I don't know if that's right. My guess is if we deep dove on that, what we would find out is that there's some kind of guesstimate in there.
Fudge factor of some
But
sort.
there's certainly a lot of living things that we don't see with our eyes.
All right. How about they carry out as much photosynthesis as all land plants combined?
I think so.
Do the students like that?
Yeah, it's 50-50.
They don't believe that, but it's absolutely true. They don't know who Prochlorococcus is.
I love Seniococcus.
And I say, here are two organisms that are really necessary for life that you've never heard of.
Mm-hmm.
I think that is amazing because most of us are raised to think that plants do it all, right? And so the students should be amazed by that.
And I've found that a lot of students come to me, and I'm not pointing fingers about this. They're unaware that chloroplasts and mitochondria were at one time bacteria. Mm-hmm. Well, I'm not putting that as well as I could for an evolutionist, but you know what I mean.
We get it, yes.
And they're kind of surprised. So that's when I shout that we need to free the organelles, that kind of thing. Elio likes that, by the way.
Free the organelles.
All right.
He's very much into symbiotes.
Evolution, anything there?
Oh, why talk about Brother Carl there? And that's a big deal
That's
to
Carl
me.
Woese, for those of you who don't know who Brother Carl is.
Oh, well, he's Brother Carl as far as I'm concerned. I really, really enjoy showing students the old ASM video about Carl Woese, and I asked them to do a word cloud of what they think of the name before and after they watch that video, and I've gotten some really remarkable responses. And the reason I think word clouds are important is that sticks in their minds.
Oh, yeah.
All right, Brother Carl, the 3 domains of life.
3 domains of life. Because, and this is something I fight with, I'm getting some students who come to me knowing what archaea are, but they think they're all extremophiles.
Right.
No.
Right. That's many people do think that.
Right.
Yes. Yeah.
Well, that's only because they were the first ones that were talked about.
Mm-hmm. Yeah.
But do you tell the students in your class that a third of them are making methane?
Yeah.
And they look at their neighbor next to them, and they can do that experiment in a big lecture hall. Look to your left, look to your right. One of you is making methane.
That's right.
Then we have microbial sociology.
Oh, I love sociomicrobiology. The idea of the microbes interacting, I've been pushing them, and I'm glad that some of the introductory textbooks are starting to talk about simplistic versions of quorum sensing. Mm-hmm. Mm-hmm. And I think it's so important for them to see that microbes are talking to each other. What is it, the old Joshua Lederberg comment that microbes are talking to each other all the time, we just don't know the frequency? Well, now we do know some of it.
Well, here's a funny story I learned here today at the meeting. I was talking to a friend of mine, Mike Cox, who runs Anaerobe Systems, and he always, even though he runs a company, he's always had a side project. So his newest interest is Clostridia fixing nitrogen. Did you know that a plant puts in an order for so many units of nitrogen fertilizer, they send a signal to the bacterium, Clostridia, please make for me so many units of nitrogen fertilizer, and Clostridia sends it back.
What do they get in return?
Sugar.
Wonderful.
I mean, this is truly inter-kingdom communication writ large, and it's transforming the way we think about dumping massive amounts of fertilizer into the environment. You just think about it.
Yeah,
you get your plant to tell the microbes in the soil, hey, we need fertilizer, folks. Makes me some. And they do.
There was a session today, the social life of viruses. They're trying to jump on this bandwagon, the virologists. right?
Right.
Now there's a whole growing field that viruses are interacting in some way.
I think it's extremely likely. And it's funny. I make a joke of it in class that scientific research proceeds by 3 stages. First is that's a really dumb idea. You shouldn't waste my time with it. Stage 2 is it might work a little bit in the laboratory, but it's not relevant to the natural world. Stage 3 is I said it was a good idea all along. And one of my students, Micheline Wong, back from my days at Occidental College, came up with the 4th one, which is, and I came up with the idea first.
That's right.
And
we've
all seen that. And this is a good thing that things are constantly, sure, it's frustrating when people are kind of hypocritical about it. But, I mean, you have to keep your mind open. I mean, things are changing all the time. That's what's so exciting about microbiology. We find out new things all the time. I'm not surprised that viruses are talking to each other.
No.
We just don't know the frequency, right?
Yeah.
It's the frequency. And, we're worried about...
What's the frequency, Kenneth? That's what he was referring to, right?
Yeah.
We just didn't know it.
Yeah. And we're worried about interplanetary travel and, aliens coming to abduct Mark.
Too late.
Too late. We have the microbes literally communicating to us now.
All right, then we have cellular microbiology.
Now, this is a term, cellular microbiology, that I just adore because it doesn't mean what students initially think. And it's fascinating to remind them by looking at microbes that can interact with eukaryotic cells. Looking at those interactions tells us how the eukaryotic cells do things. And you can actually see the light go on when they get that. And it's so rewarding to see that. For example, learning about how lysosomal fusion takes place because of bacteria that inhibit that process, how actin rockets form. First off, they love actin rockets. Who wouldn't?
I mean, actin rockets are very cool in how they facilitate intercellular movement.
Mm-hmm. But understanding how actin is recruited as part of normal processes. I even read that I believe there's now even they're finding there's one, and I don't know the organism, but there is some eukaryote where they have bacterial symbionts in the endoplasmic reticulum. It was one of the things that we were talking about with a group of people today, and that will give us a lot of insights into how vesicles travel. So,
vesicular trafficking is probably invented by the prokaryotes.
Well, everything is. Right?
Well, they're here longer.
But that's my point, is that particular key really helps them understand that you can use microbes as a tool. I mean, why do we know how well ribosomes work, or how ribosomes work at all, I should say? It's because of antibiotics.
Absolutely.
Yep.
And
it's a tool.
Yeah.
And the last one is microbial cell structure, which is subtitled The Age of Imaging.
Yes.
And this is all about power of microscopy, right?
And not the microscopy that I was raised with.
No. This is spectacular microscopy.
Right.
The opening session at this microbe meeting, the second talk of the evening, was spectacularly filled with images. And she did a remarkable job, and hopefully ASM will post that video by the time this show goes up, and so we can link it potentially in the show notes.
I cannot express how electron cryotomography has transformed the way that I think about bacterial structures, let alone eukaryotic ones, or nanosims, where you can actually measure metabolite flow between adjoining cells, and I mean bacterial cells, cyanobacteria in particular, between the heterocyst and the vegetative cells, is fabulous. Now, of course, every student then says, do we have one of those machines? And I'll say, no, we don't. But that's the important thing about the world we're in now. In a few years, we all will.
Well, we do have one of those machines. It's called the open ocean.
All right, now we have Thor.
Thor!
This is a very important paper to me.
This is not the guy with the hammer.
I would say this is going to be tough for students to appreciate, but tell us what you do with it.
So
what is Thor, and what do you do with it?
Well, this is the thing. I have been fortunate enough to know Jo Handelsman for a number of years, and there were some very unusual results that she had in the lab, and some of the things that she had published on related to them got me thinking. And I pestered her, and she's very tall. And Joe, if you're listening, thank you for being kind about it. Because have you had an idea in your head, Vincent, you couldn't get out? So this is what she found out.
Podcasting.
Podcasting.
I'm afraid to ask Michael that question, come to think of it. But yeah, look at the face. So here's what happened. A situation was when folks in her lab were looking at Bacillus cereus, which is commonly found in soil around soybean roots. There are a lot of good reasons to be interested in this particular strain of Bacillus cereus. But long story short, some of the isolate, and they had been single colony isolated 3 times. Now you and I think that's good enough for everything. But then the student kept those plates in the cold room for a couple of weeks.
Oops, something happened.
And those single colonies had other things associated with them. And I was thinking about Bacillus cereus. Now, it turns out it's a flavobacterium, and it turns out that it's actually able to consume peptidoglycan. Now, Bacillus cereus, as a very interesting gram-positive, has, I couldn't predict, 50 layers of peptidoglycan?
At least. It's very, very thick.
Right.
It's thicker than a roach.
So I was saying to Joe, I imagine that these are hitchhikers. And it's interesting that you carry it through 3 colony isolations and about, I think, 5 to 10% of them when they do this. Yeah. And so, what it stands for is the hitchhikers of the rhizosphere. And that's what Thor stands for. Now,
and did she borrow your word, hitchhiker?
She has said that to me, but I do not wish to take credit for things that everyone does great work on. My name is not on this paper, I would say, so I don't want to give it a false impression. I used to call these flavobacteria peptidoglycan surfers. Sounds like a new wave band, but I do this. This is something that's been in my head because every time I look at these large merithrix, for example, that I see in Puget Sound, these are bacteria, but they're huge, and you see all other kinds of organisms attached to them. So it wouldn't surprise me. What's interesting about the flavobacterium is that it clearly is interactive. It's making enzymes that are able to consume the peptidoglycan.
But not kill the host. We also know examples from Margaret McFall-Nye's work that those fragments of peptidoglycan can be signals. And that turns out that's the signal for pertussis, right? No. Is it pertussis? No.
Tracheal cytotoxin.
Yeah, it is pertussis.
Yeah, but that same segment of peptidoglycan will induce Euprymna scalopes, the Hawaiian bobtail squid, to form a light organ, which shows you that these little bits, this is where we come up with the idea of talking about microbial associated molecular patterns as being important. And that interaction is what's central to this paper. Do I think that students can automatically understand the details of the paper? What I like about this paper is there's a level of it for first-year students, and then there's a level of it for juniors and seniors because I'll be teaching from it. And I would draw – so they have themselves basically 3 members that they're talking about, and they're all interactive.
And they come from roots.
They come from roots.
Right.
And they're clearly playing with one another, inhibiting one another, creating a broad series of types of interactions. And I'm really pleased with the flow of the paper. I certainly look at interaction maps, as you'll see, for example, in Figure 1, network analysis, and I feel very inadequate when I look at this because it's not –
But this generation of student is into concept mapping.
Yes.
And so this is almost, if you will, second nature to many of them because in their middle school and high school years, they have been doing concept mapping, which is nothing more than what Dr. Handelsman and her crew effectively are doing with this networking story.
And I would say I was trained as a geneticist where we look at a single gene and a single phenotype, but that's not how nature works at all.
Oh, no.
And so when you start talking about systems biology, it's one of those moving targets for definition. And that's one of the things I really like about this paper because now you're looking not one organism and one condition, but 3 different organisms that act in different ways. It is absolutely true that Bacillus cereus acts in different ways around the various members. Some of those changes are eliminated by putting another member of the 3 in. For example, if you look at figure 4, there's some wonderful bits about how Bacillus cereus will actually form these dendritic forms in the presence of some microbes, but not others.
Right.
So the complexity of the story is huge. As Dr. Arnold last night in her talk to the audience here at Microbe said, once you get into combinations of 3, you exceed the national debt of the United States many-fold, and you have 35 trillion different possibilities from just changing 3 things. And these microbial communities are much more complex than 3. So this is a good place to start to really cement the wow.
And the materials and methods section is exceptionally clear. You can see exactly how they're doing things, and it's not as complicated as a student might find other types of papers to be, where there's a lot of work done with filter-sterilized supernatants.
Yeah.
There's a lot of it about using low and high concentrations of nutrients. So it's a wonderful test bed for interactions between organisms. Because if you think about it, that's the real question in life.
Yeah, and they can imagine how they could participate in designing an experiment to ask a question.
Yes.
And that's what you want them to do, is to ask a question that they could get an answer that they could heft in their hand without complex machinery, fancy budgets, or anything.
What's the old paper, Toothpicks and Logic?
Yes.
Mm-hmm. Right. The idea being that, we get carried away with giant machines. And I often say to students that the most effective machine for analysis of science is between their ears.
Yeah.
The best visualizer actually is their eyes. And the old joke that Isaac Asimov made is that discovery happens not when you have a damp Greek leaping from a bathtub saying Eureka, bowing to Archimedes there for you.
Yes.
But in fact, when someone says, and I've done this, and I'm sure you have too, where you kind of look at something and go, that's funny, isn't it? That looks weird. And that's where it starts to happen.
My old advisor, Walt Koneska, when I would bring in data sets that were puzzling me and try to explain them to me, he would just respond to me, it bothers me. Mm-hmm. And it bothers me. And what do you do with that?
Well, and it bothered me with this.
Yeah.
Because I thought to myself, how is the flavobacterium living? And I still don't know whether it's possible to do a section through the Bacillus cereus and see if they're burrowing into the peptidoglycan. I don't know.
Mm-hmm. Maybe. And remember, it's the molecular corset. It's forming this girdle, because Arthur Koch, who's the man who taught me most of what I know about peptidoglycan, he was always intrigued by it. And if you think about it, think about the square bacteria.
Oh.
How do you make a square bacterium and get it to divide via binary fission? And, did you get a square out of binary fission? I mean, Arthur would torture us poor graduate students with these concepts, and you begin to think about it and the wonder of peptidoglycan. And we don't have a zebrafish model that's transparent that we could watch this, because the resolution is not yet there with our computer tomography.
Yet.
Yeah.
And that's one of the things about Elio's paper, again, or essay, that's so important. So just to summarize the Thor paper, because you're going to tell me to move on, right?
I would like you to summarize it, yes.
Yeah. I think that there is something that's very basic that I don't mean any negativity, that a first-year student can get out of this, and that has to do with the specific type of interactions between even 3 organisms. Mm-hmm. One thing that they talked about in the paper in particular, the authors discussed, was sliding motility.
Oh, my.
Mm-hmm.
And that's a wonderful business. It's actually quite relevant to some things that interest me. The idea that organisms can develop surfactants, or, as I say, a slip and slide, to move around. And they're not used to thinking that way. And then I take them from that and say, what does that mean in soil? Which is the important part. So there is certainly a level where a first-year student can look at some of these interactions and can even think about taking a culture supernatant and adding it or plating them together. And I think that level makes sense. And in fact, I'd like to do that with first-year students, because there is nothing that works better than them seeing something really interesting on a plate.
So what are your thoughts about a lot of our undergraduate curricula are dispensing with hands-on laboratories and inquiry-based laboratories because of the cost often associated with it, the time, and they're effectively value engineering them out for computer simulations or things along those lines?
I would die on that hill, and that's the second paper.
So I set him up for a transition.
But before you go, I just want to say, so Thor, we'll put links to all these papers in the show notes. 3 bacteria from soybean roots selected because they interact in certain ways. And they say it lovely. Thor has 2 properties, colony expansion and biofilm formation, that could not be predicted from the individual behaviors.
Of one.
And so now you have 3 genetically tractable organisms. You have the genome sequence. You can start to say what is controlling this, right?
You can start to dissect it.
Exactly.
And I want to point out the term that they use is one I like discussing in class, emergent properties.
Yes, exactly. Emergent. Yes.
And what I often tell people is, what if I told you here's a metal that's explosive and a gas that will kill you dead in seconds that you use every day when they're mixed?
Yep.
And that would be table salt.
Yep.
Right? And that's an example of an emergent property.
That's right. Exactly.
And in this sense, they didn't expect to see some of the things that they did.
Okay. Next paper is another cool one. It's all about phage therapy.
And I have to say, as we start, that there's a new book out by Stephanie Strathdy and Tom Patterson called The Perfect Predator, which is the discussion of a similar situation having to do with the use of bacteriophages to treat disease, multiply resistant disease.
That, of course, is a book for the general population.
And a TED Talk.
And this is a scientific paper in a journal, so a little bit different.
Oh, but it's fabulous, isn't it?
This is Nature Medicine, Engineered Bacteriophages for Treatment of a Patient with Disseminated Drug-Resistant Mycobacterium abscessus. This just came out, and I hadn't even seen it, so thank you, because we're going to do this probably on TWiM.
Yeah,
TWiM.
Absolutely.
TWiM. You want to do this on TWiM?
Yeah.
It's got viruses in it.
That's okay. They're phage.
Okay. It could be one or the other.
Such a hater.
No, I'm not a hater. I want good science.
What's good in this for students?
So often when we talk about bacteriophages or we talk about what we learn, students are kind of drifting off into nowhere land, especially first-year students. I find that juniors and seniors are really interested in the ideas of phage therapy, which have been around for a long time, but they didn't work very well. What's remarkable here are a couple of things. The first is the fact that it's using undergraduate research, which takes me to what I was just discussing with Michael. When you take away, I mean, this is an example of one of those.
That's why you're going to die on this hill.
That's well, yes.
I'd die on that hill with you.
So it shows that, this idea that, it's just showing people things. This kind of inquiry-based learning creates this whole library of bacteriophages against a mycobacterium that doesn't cause disease, Smegmatis, right? And I know for a fact that a lot of the folks working on this, the great Graham Hatfull, were thinking, well, this might be appropriate for tuberculosis, but not so much, was it? They were able to make it work in this case and really work towards saving a life.
So these are high school programs where kids get M. smegmatis, and they go into the dirt and isolate phages that infect.
That's correct. And you'll notice some of the names are unusual because every discoverer gets to name them whatever they want.
That's
which is fine by me.
And here was a 15-year-old kid with cystic fibrosis, had an awful infection. They had to give him a lung transplant. He was infected with M. abscessus. They couldn't give him antibiotics. It made him terribly sick.
Right.
And they came up with the idea to give him these phages.
In a cocktail, 3 of them.
And did we know that those phages would actually infect abscessus to begin with?
So that's what they did. They isolated a strain of that organism from the patient.
Right.
And they were able to go through, and I'd be honest with you, with a life like this, I would, I have never done anything in my life research-wise where someone's life is in the balance, right? So it would be very hard on me. But they had thousands and thousands of different phage isolates to work through. And they did. And for example, they found one was clearly lytic in terms of plaque morphology, taking you back to the P word. And in fact, they've ones that were they're more turbid. Then they say, well, since we know the genomes of all these, and they do, we'll just go engineer it.
It's amazing.
Remove the repressors, make them lytic.
They removed the repressor.
That makes sense. You can do, and that you can engineer a phage to do what you want is remarkable.
I mean, I want to remind you, we're looking at, I want to read this sentence. The name of the phage is Zoe J, so I'm sorry about that.
Poor Zoe.
Well, no, this is good.
A high school student.
She named high school, so she named it after herself.
Or it might be an undergrad, but this principle still, because this is usually a first-year class.
Okay.
A lytic derivative of Zoe J was engineered using bacteriophage recombinering of electroporated DNA, bread, to precisely remove its repressor gene. This effectively infects and kills GD01, which is and isn't that a wonderful,
isolate for the patient, right?
Yes.
It's amazing.
Yes.
No, it's amazing.
Yes.
But then they went back and did more screening, right?
Yes.
Of more samples. And they ended up with, what, a cocktail of 3?
3. Yeah.
Which they then gave, remind me how they gave this to, let's see. They do some killing in vitro to check that it's killing.
Right.
And then they gave him IV therapy.
Right.
10 to the 9th PFU.
Small number.
Every 12 hours for 32 weeks.
That's a lot. But remember, Mycobacterium grows slowly. And the way phage work is you need an actively growing cell in order for it to effectively take things over. And so that's probably the reason they used such a long period of time.
It's amazing. It's just remarkable. And the kid got better.
Yeah. They were also applying it topically to some areas too.
He had lesions all over.
Right.
Right, from the mycobacteria.
So it's really important to emphasize that what was a Howard Hughes Medical Institute funded project for high schoolers and undergraduates, the idea of teaching science in a way that is interesting to students and directed. It was a 2-semester kind of course. I wish I could teach a course like this.
You can.
Oh, no. Well, then I would have to not teach anything else ever. But what is wonderful about this is that they go through the process of isolating, they go through the process of characterizing, they do EM, they get a genome, and they've worked out all the kinks. And so because of this, now we have a wonderful resource for anything involved with mycobacterial infections, in my opinion. And I mean, this idea could be used with almost anything else.
But who made the leap to say that these phages would infect abscesses? Is that something obvious that you would assume?
They had thought at the time that what they were doing with Mycobacterium smegmatis might apply to tuberculosis. And why not try to see if it would work with this?
But TB is a level 3 pathogen, so you need special training and special laboratory equipment in order to go. But smegmatis is a level 2 organism that you can generally get away with, exposing undergraduates to without much fear of biocontainment and other issues.
And despite the level of this that might not be for a first-year student, I mean, certainly we can say removing a repressor without going into how that's done. That's understandable.
Yeah.
Because they get a certain degree of gene regulation in first-year courses from bacteria.
Many of them are learning about the lac operon in high school.
Yeah. But notice in this case, it's almost a narrative for the treatment.
Yes.
It's almost like reading like a thriller or a report on what's taking place. And since many students have an interest in medicine, here's a life being saved.
When I read this paper, I thought of the movie script.
Yeah.
This is actually a movie script. If you read this and you literally see the high school kids developing this, working at it, and you make the star of the show the poor kid with the abscessus infection, and you can see it's got all the elements of a good movie.
The book you just mentioned remind us that
The Perfect Predator.
Isn't that going to be made into something?
I wouldn't know. I have heard rumblings, but I'm in the mushroom core in many ways, kept in the dark.
Mushroom core. You're in the Lincoln log.
First therapeutic use of phages for a human mycobacterial infection,
and the first use of engineered phages.
Amazing.
Isn't that wonderful?
That's not necessarily true. We engineered phages 20 years ago, and we used them to treat
people.
No, because we had to worry about the FDA.
All right. Well, that's what they did here, right? Which is remarkable that the FDA let them do this because they have to run it by the FDA, right?
They have to run it by the FDA.
So I have a couple of questions about this, and I don't want to rain on the parade. But one thing that always worried me is when you have a phage lysate, you're going to have, I mean, how pure can you get that phage lysate? I used to do, cesium chloride gradients or sucrose gradients to isolate phages. I'm worried about things like LPS, which is not a problem from a gram-positive, I realize.
But there are reactive immunological molecules associated with it. That's the principal rub with the phage cocktail because it will set off the immune system because you're given parts.
And it doesn't look like that happened here. They actually talk a little bit about it. And with weak cytokine responses. So, this is going to be a moving target to some degree.
10 to the 9 is not that many phage. So it was probably diluted. And so you're diluting out the dead body effect of the phage parts or the bacterial parts involved in the construction of the phage. Once upon a time when I was in the phage business, that was the rub, is getting it to a level that you could make a ethical pharmaceutical.
We did a paper earlier from Paul. What's his name? Remind me. He gave a talk in the Phages in the Wild session today. All right, TWiM 44, and there they purified it to get rid of the endotoxin.
Right.
His name is Paul Turner.
Of course, yeah. Oh,
they treated a man who had a aortic implant, Dacron, right? It got infected with Pseudomonas, and they poured phage. His chest was open because it wouldn't heal.
Yes,
they poured it right on the graft, and he got better.
Isn't that amazing?
And then when the day the paper came out, he had a heart attack and died. Isn't that sad?
Ugh.
But they purified away the endotoxin because he couldn't tolerate it.
Yeah,
right?
and that's the thing I would think about. There's also a lot of news now about bacteria inside human cells, inside tumor cells that might have various effects. So once again, it takes us back to sociomicrobiology.
Jeff Hasty. He's right here at UCSD. Well, right here, 8 hours away.
By train.
Bacteria can get into tumors and they kill them. It's amazing.
Or I've heard rumors that they can get inside tumor cells and protect those tumor cells from chemotherapeutic agents.
Is that right?
This would not surprise me either.
Well, that's not good.
Well, it's the doctrine of microbial infallibility.
Yes.
All right.
There has not been one compound that man has designed that microbes can't figure out.
And I want to say, before we move on, this is so important under acknowledgments for this really important paper, thanking the students that found the phages. Now, I love telling students that Taq polymerase is all because of an undergraduate student working with Tom Brock, and that's one thing. But look at this. This is a wonderful bit. Their work that they did for a class is now something to be used in medicine.
All right, your next paper, Mega. So we go from Thor to Mega, right?
What are you looking at now?
Your show notes.
Your show notes. Michael Baum's Mega Plate Approach.
Oh, so we don't have a paper for that, but what's wonderful is a YouTube video, which I'm going to give you a link to. Is that in there? And so what's wonderful about this is these are the giant plate that Michael Baum has and this is based on Roy Kashoni's work. Michael Baum has perfected a lot of it and carried it further. So what you have are stepwise different sections of the plate with different amounts by a factor of 10 of antibiotics. Mm-hmm. So you have none of them at the outer edges, and you go up toward the center where you have quite a high concentration.
That's a paper covering that topic in Science.
Oh, yeah, right.
And
there you go.
There it is. And what I like about this is that you can see in the video, you can watch those individual mutational events that lead to a bloom as the microbes are able to grow at the higher concentration, and then the next.
Oh, it's a wow. I mean, you just watch that video and you say, oh my, how are we able to use antibiotics at all?
And what I like about it, I've used that with first-year students to grab them. And what I like about the particular video that Michael Bay made is there's a really good description of what's taking place. Now, later on, it goes into kind of systems analysis, which I don't think is going to hit a first-year student.
It doesn't have the wow.
But that is a wow.
Oh, but if you're doing this where you have both biology students as well as the computationalists in the room, they will then bring their toolbox to the table, and that's where a biologist and a computationalist get together and figure out the next great thing.
I really want there to be a discussion and a lab, and I want it to be a lab that isn't like a demonstration.
No demonstrations. Inquiry.
Showing antibiotic resistance and how it works. And the closest I come to that is if I do a transformation experiment with, please forgive me, it's called PGLO. It's a plasmid that carries, it's ampicillin resistance gene, beta-lactamase, and it has a GFP marker. And so when you shine UV light on the transformants, they,
glow in the dark.
But then you wait 2 or 3 days and you get satellite colonies, because the way that beta-lactamase works is it's secreted by the transformant. And you're reminding students that there aren't really dead cells on that plate.
They're just waiting.
And then you get all these small colonies around it, but they don't glow under UV because they don't have a plasmid. And I love turning students, my first-year students, loose on that. Tell me what happens.
It's great.
I do have to tell them that if I take the central colony and streak it out on ampicillin, it grows, and I take the satellite colonies and they don't. I've got to tell them that.
Well, that gives them the whole notion of how does it breed true.
Yeah.
That teaches them what that phrase breed true actually means.
So it's a wonderful video to show.
Yeah, well, being a video is perfect.
Yes.
Right? Because that's what they're growing up on. And then to have science as part of it, teaching them something.
And it's relevant to the real world around them.
Yeah.
The rise of, and I spend a lot of time saying that pathogens are a small minority of the microbial world, and that's true. And I'm not even making a joke about human narcissism. I am concerned about the rise of drug-resistant pathogens. And
as we should be.
Yes.
All right, next we have a cool kind of This Has Been My Life by Roberto Coulter, Biofilms in Lab and Nature, a molecular geneticist voyage to microbial ecology. It's kind of a story of his movement through biofilms, right?
It's a wonderful story, and it's kind of personal to me, because I knew Roberto many years ago when I was in graduate school. He was a postdoctoral student in Charlie Yanofsky's lab, and I was a beginning graduate student who was petrified of being in that lab.
Of Roberto or Charlie?
Yes. Well, no, I'm not being negative. They're so brilliant. They do such great work. And I mean, it's been so wonderful to watch the arc of Roberto's career and the wonderful things that he's done repeatedly.
For those of you who may not know the career of Charlie Yanofsky, just think of the tripoperon and attenuation, and you truly see the wonders. And this was all done before the age of computers and the topology of RNA was figured out at the coffee shop, staring at DNA sequences and folding the RNA sequences, and it's a fascinating story.
Mark, I love the epiphany of the fish tank, where he claims to have discovered biofilms by looking at a fish tank. The water was clear, but the walls were getting cloudy.
Yes.
Right? A fish tank that his students gave him, they put it in his office because he was depressed, and they said, you should look at this.
Look at the fish.
Well, Charlie was a big aquarium aficionado.
Really?
Yeah,
I didn't know that.
This is a wonderful, wonderful paper because of how well it's written, how accessible it is even to beginning students. And in particular, Figure 1 is a big deal to me, because one of the things that I try and teach students is that what we mostly see in the lab are lab rats. And so when you look at wild Bacillus subtilis, it's very different than the lab strains of Bacillus subtilis. And if you look, it's beautiful, the pellicles that you see on the liquid surface, and there's quite a lovely I'm trying to figure out who took this picture. I know Scott Chmielewski does a lot of great photography, but this might have been before his time. But it's wonderful because you ask them, what do you think all of these ridges are? Why are they there? And you notice, and this might be something for more advanced students, he tries to address that a little bit in figure 3. And you'll see there are different subtypes of cells you get in different places, similar to the work that Travisano and Von Cooper have done based on Paul Rainey's work with the diversification of Pseudomonas fluorescens in pellicles. And cheaters that are taking advantage of being next to the air and all the rest.
And for those of the listeners who may not know what a pellicle is, you can do this experiment at home with a coffee cup and cream. You just leave your coffee cup sit in a cool, dark corner, and a pellicle will form on it, and you will say, "Oh my, what's going on in the corner?" And you'll get a nice pellicle.
Do you use this to introduce them to biofilms?
Yes,
because it's accessible and it's
you don't have a fish tank.
Well, I have a story about that, but we don't have time necessarily.
That's right. We have no time.
But it is interesting to note that surfaces are where the action is always. And it makes sense, right? If you're just going with the turbulent flow, you're not going to get as many nutrients when the world comes by that you can reach out and nab.
I met a guy in Albany a few months ago who is studying the biofilms on your teeth.
Oh, yes.
Gets pieces of plaque and does microscopy. And he stains with reagents. I think it's nucleic acid stains that are specific for the ribosomal RNA of different bacteria. And you can see the complex communities that are making up these films. It's amazing.
And you can have students do this experiment in class. You segregate the class into a population that doesn't brush their teeth, and you pick a time, a weekend, 4 or 5 days, and you ask them, can you feel the fur growing on your teeth? And they can. And you can do another experiment with sugared beverages where you paint the sugared beverage on their major tooth surface, and then the biofilm will actually form a starch. And we all learned how to stain starch in middle school with iodine. You can put a little drop of iodine. You can see the bacterial starches, the bacteria that are accumulating the starches on the tooth surface. So you can do the control on the other side where you paint a diet beverage or you don't feed them.
Have you seen Carl Zimmer wrote a wonderful essay about microbial ecology, comparing it to what people knew about lakes. And I'll have to get you that information. And it does talk about the interactions between the different microbes in our mouth. So you can imagine you're alone on a Saturday night, but there's a cacophony of communication taking place in and on you all the time. You're never really alone.
You just put your finger in your mouth and smell.
Oh, please.
All right. We next have a wonderful perspective in PLOS biology, life in a world without microbes.
Oh.
Gilbert and Neufeld. And I love this. It goes through the thought, which is what Pasteur said, life would not remain possible in the absence of microbes. And they say, well, really, would it or not?
So there's actually...
Now without oxygen, we wouldn't have had oxygen.
There's actually a video of this that's been put together, which I'll send you a link to.
Please.
But I honestly like the written version a little bit more. Mm-hmm. And you notice that there's
the book is always better than the film.
But you'll notice that what you'll see is that there's a lot of business talking about Elio's blog.
Yes.
And a lot of discussion about someone who'd made comments on there. And it's the idea of bringing new ideas together. When you deal with the fact that most students think of bacteria, microbes as, quote, germs and therefore terrible, unlike the talk I gave earlier, what happens is that they don't appreciate all the different things that they do. And this isn't like a laundry list of why microbes are great, but the air we breathe, the nitrogen we need to drive things, it's all there. And it's a microbial planet.
This is great because they go through, let's say we take the bacteria and the archaea alone out, and life would be pretty bad, but it would go on for a while. And I love this part. What about the waste? What happens to the accumulating waste?
Especially if we just eliminate the fungi.
And then, of course, if you take out all microbial life, they said then we're really screwed. But I love this part where, they say humans would not be able to deal with not having microbes really, really quickly. We could make nitrogen by the
Haber process,
but, recycling of nutrients would be problematic. We just would not be able to keep up. And they say something here that social chaos would ensue rapidly.
Oh, yeah.
Right? which is true. People will start killing each other to get food.
Running to the shores where there might be a source of nitrogen, right?
Yeah, well, they say that the shores, the coastlines, life would distribute on the coasts where nitrogen-rich fish could be harvested and nitrogen scavenged from seawater.
But remember, the fish are making their living from the microbes as well. They're often eating plankton.
Yeah.
I just have to laugh. There's that wonderful bit at the end with the little asterisk about whether or not we should consider, mitochondria and chloroplasts as being bacteria, because if they vanish,
boom,
yeah,
death would be immediate in a minute.
So it's a really good reminder, again, to introductory students of the centricity of the microbial world. And, I get told all the time that I preach it, but that doesn't mean it's not true.
That is indeed correct.
Right.
All right, we have two more, and they're just amazing. We have a reduction of cholera in Bangladeshi villages by simple filtration. This is such a beautifully written paper.
Isn't it?
It's from 2002.
And it really, really works, folks.
2003.
This is not science fiction. This is science fact.
So the thing that's so cool about this paper to me is it reminds everybody that it's, they have this idea that microbes are just swimming around looking to colonize you and cause disease. But as you find out, there's a taxi cab that they're riding.
Yeah, they're out Ubering.
They're Ubering on copepods.
Yeah.
And those are, I'm not going to call them macroscopic, but they're certainly large enough that 4 folds of sari cloth actually can reduce by 2 logs.
Just enough to drop the infectious dose.
There you go.
Because dose matters.
And what I like about it is that it's real science being done in a place that needs help, where it's not expensive. It doesn't involve machines that go ping. So it's a wonderful, wonderful example of socially relevant science that I found most people forgot about, and I like to show it to them. Besides, Rita's wonderful, so I like doing that bit. And it's not here, but there are some wonderful photographs of copepods that are just covered. And since many of
Covered
them,
with Vibrio, right?
that many of them use chitin, I'm wondering, once again, it's this matter of communicate about interacting with one another.
It could go back to your story of the flavobacterium riding on the bacillus.
That's right.
And what the Vibrio are doing is riding on the chitin of the copepod.
And there's even more stuff here. There was a discussion about keeping out other small crustaceans that are responsible for guinea worm.
That's right. Drunculiasis, right? You can filter that out as well from drinking water.
Rather than boiling, which you can't always do, but would work.
It's expensive.
In terms, and it's not carbon neutral.
It's not carbon neutral.
Well, they say they can't afford it. Building fires to boil water is simply not possible.
Yeah. But what I like here is this isn't a bunch of well-to-do Europeans saying, here's a wonderful machine that you can do.
Buy it. We'll give it to you.
Right.
The Gates Foundation doesn't have to go out and spend billions of dollars delivering it.
And I'm glad that the Gates Foundation does things like that. But what I am trying to say is that here's something that fits within the culture where people are going to be comfortable with what they're doing.
Oh, that was important because they said you could use nylon, but these individuals would rather use sari cloth.
Why not?
A few people didn't want to use it for whatever reasons, but they said it's acceptable in the village. I think that's just wonderful. They also say, we suggest that a simple solution to a global problem can be achieved when the ecological basis of the disease transmission and its reservoir are known. I think that's great for students, right?
They have to understand
Really,
putting
truly.
2 and 2 together gives them a solution. And it's a really profound solution because cholera is a bad disease.
Yes.
Despite this simple approach, it is a beautifully designed study. They had control groups. They taught people how to wash the sari afterwards, right? And they had different villages do different things. Control in one village, sari here, because if they did neighboring houses randomized, they would tell each other, I'm filtering and you're not, and that would screw up the experiment. Isn't that beautiful?
Because it has to be done that way to find out what works best.
Yes, yes.
And so I'm really pleased because it's too often, and I don't want to kind of allude and kind of like devolve into politics, but I'm not happy with a person in one culture going to another and telling them what to do. This was respectful of different ways of thinking and knowing things. And it was delightful to see. And I remind you, that's 2002.
Yeah.
Almost 20 years ago.
Yeah.
It's remarkable.
You imagine how you would feel if you had helped do this work, the difference you would have made in lives.
Oh, yeah.
Well, the people involved in the study probably feel pretty good about it, right? Right? So,
Well, more
it's
importantly,
wonderful.
the agency that funded it should be especially proud.
We should do this on TWiM, actually.
We should.
Because it's a classic, right?
Yeah, it is. And we haven't been doing enough of the classic papers. And that's effectively what Mark has brought to us today, is how to inspire the next
These are
generation.
great papers. One more. PLOS Biology, the placenta goes viral.
That's for you.
Retroviruses control gene expression in pregnancy. I have to tell you, Mark, I write a blog post every week, okay, on Virology blog. And I read this on the plane yesterday, and I wrote a blog post last night about it because it's perfect. I love the syncytium story. And then this part with the corticotropin-releasing hormone I hadn't known because it was just published recently. So this is wonderful.
Doesn't it seem like science fiction? I mean, when you...
So you gave Vincent the wow.
I gave
Totally,
Vincent the wow.
Totally. I mean, even I, a jaded old person like myself, can use wow. And
OMG.
In fact, I do wows all the time. I'm amazed at science to this day.
It's beautiful.
So what is this? What's good about this for your students?
Well, a lot of times when we talk about, I mean, I try to do things like point out how much of their genome is viral. And I don't know how well that really works. But when you start to talk about how the placenta works, I mean, the way I introduce this usually is I'll talk about cell fusion experiments that were necessary to show, how membrane proteins move across the surface, and usually some student will very wisely ask me, so why would you fuse things together? And I said, well, of course, many viruses want to have a larger kind of biomass to make more, viruses to virions to escape completed. And then I'll tell them, but the reason we're here is because of that kind of thing. And they go, what do you mean? And then we talk about the placenta and we talk about syncytin. And it's really interesting. They don't know much about endogenous retroviruses, of course, in my courses because I don't teach virology. They know about HIV and they know about influenza because I kind of insist on that. But this is really good because it shows that there's a viral history to our species.
Do you ever
Not just
inspire
us.
them to read some science fiction, like Darwin's Radio by Greg Bear?
Yeah, well, I've Greg Bear he's wrote a wonderful novel about how microbes rule the planet called Vitals. Greg Bear is a fabulous writer. Actually lives in Seattle, though I don't get a chance to see him very much. What I like here is, again, people think of, and I've discussed this with you in the past, Vincent, just as many people think of all bacteria as being bad, they'll think about all viruses as being bad.
Absolutely.
Vincent's opening line to TWiM, the viruses that make you sick.
That's right.
Well, in this case, the viruses that allow us to
Breed.
be placental mammals.
Yep.
And there's a lizard that has the equivalent of, yeah, there you go ahead.
Equivalent of a placenta, where apparently the retroviral glycoprotein, which causes cells to fuse, contributes to the formation of the syncytiotrophoblasts. Yeah.
And notice the exceptionally clear diagram.
It's lovely.
Oh, yes.
So you show that to them?
Absolutely.
Yeah.
Because
very nice.
you don't have to read very much to understand that. And I really enjoyed the idea of taking essentially an enhancer sequence and co-opting it to express something else.
Absolutely.
I discuss that when I have a little talk about cancer biology, the way that some viruses can alter expression. But this is something, again, which is very important to us. And again, it does take you to Darwin's Radio in a way.
Yeah.
Oh, it's beautiful. So there are two stories: syncytin, a co-opted retroviral glycoprotein, and the promoter for this hormone that controls birth timing. The cool experiment here, they put the human gene into mice, and mice don't make this in the placenta. And the mice give birth later.
It slows it down.
And when they knock out the viral sequence, it goes back to normal, showing that the LTR, the viral promoter enhancer, actually is important. It's really beautiful.
And it really does demonstrate how much we're all related when you think about it, because you can do that kind of work in mice. And you can see those. It's like when people would take ultrabithorax and Drosophila, and they would put that in mice, and then we'd extra ribs.
Right.
It's a wonderful example of the unity of life, yes.
So do they, in the end, get this idea that we have viral sequences in us and that they're actually contributing to us?
That's what I've been trying to push.
Yeah.
The idea of it, and I really don't like the term junk DNA because I don't believe it's junk. I'm just not smart enough. I'm just not smart enough to know what it is. But in this case, there's definitely something which is integral to what we are as a species. Two different ways.
Yeah.
That's what I like. One discussion.
And I'm sure there are many more examples to be found, right?
Or ones we haven't found yet.
Yes, for sure. Oh, these are just two that have been well worked out. It's lovely. Well, that's been fabulous. Thank you.
I really think that it's easy. You're very welcome, of course, but it's very easy for people that love microbiology the way that we do to just fall in love with a complex paper. I mean, when I talk about attenuation, for example, I'll go crazy on that subject, and the students are frightened. Or when you start to talk about the way sigma factors form cascades for sporulation in bacillus, it doesn't even matter if you show, the Harvard video on how that works. It still doesn't matter. But it's important to, just as I talk a lot about the beginner's mind from Zen Buddhism, the idea of looking at things as somebody new to something. I think it's really important as educators that we remember how things look through student eyes. And what do we want them to take away? And in each of these papers, we've talked about important concepts. Not only that, level of concept that would be good for a first-year student who doesn't have much background, and stuff that could be easily dissected into more complexity by more advanced students. And I would much rather see us slowly ramp things up in a similar way. I often use very old papers when I talk to first-year students because everything is very clear-cut and they can kind of visualize how experiments are done. Are there a lot of controls in these papers? Not necessarily, but there are some that do have them. Mm-hmm. But there are important take-home lessons that I think will stick with students.
Cholera paper had a great set of controls, right?
Yeah.
Unfiltered. All right, that is TWiM. You can find it on any podcast player, and most of you do listen on a phone or a tablet. Please subscribe so you get every episode, and we learn how many people are listening. That's important to us. So please just hit the subscribe button, and even if you listen on your desktop, please subscribe. If you really like what we do, consider supporting us. You go to microbe.tv/contribute. You could give us $1 a month over at Patreon or PayPal. It's not going to make any difference, except it's going to make you smarter because you'll have not only TWiM, but TWiV, TWIP, TWevo, and Immune, 5 science podcasts, all by wonderful scientists who want to teach you about what they do and how they do it. It is a great bargain. I would say that's one of the greatest bargains on the planet Earth. $1 a month. And if you all did that, it would help us in a lot of ways, which we're
fortunate because we're visiting with Mark here at the meeting. Since Vincent and I are here, we would normally, have to go through great difficulty to get Mark to settle down with us for.
And of course, it's great in person because we can see each other and watch our gestures, and I can see your tardigrades sitting on the microphone.
There they are.
They've been very quiet.
They haven't attacked us. I was concerned.
They've done a good
job. They live in space, which always troubled me.
And, of course, if you have questions and comments, TWiM at microbe.tv. Michael Schmidt is at the Medical University of South Carolina when he's not here out in San Francisco. Thank you, Michael.
Thank you, Vincent and Mark. Great pleasure.
Thank you.
Mark Martin does not make shoes.
No.
He does not clean your clothes. He doesn't martinize. However, he's a professor at Tacoma, Washington, University of Puget Sounds, where he inspires the next generation of microbiologists. Thank you for sitting down with us. It was wonderful. I really enjoyed it.
Thank you so much.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank ASM for their support of TWiM, Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.